Hi everyone, my name is Sabrina Rondeau. I'm a PhD candidate in Nigel Raine's lab here in SES, and I study the impact of pesticide residues in soil for ground nesting bees. Today I'll present the results of one very exciting chapter of my PhD thesis that looked at the sublethal effects of pesticides used on squash crops for the Ori squash bee. In the Rain Lab, we love bees, and not just because they're cute. By pollinating crops and wild plants, bees are responsible for roughly 1 in 3 mouthfuls of food we eat and contribute to the reproduction of almost 90% of flowering plant species. But since they are important pollinators of crops, bees are commonly exposed to pesticides while foraging and nesting in agricultural environments. These pesticides are used to protect crops against pests and diseases, but it can also have multiple detrimental effects on bees and other pollinators. In particular, one group of insecticides, the neonicotinoids, has increasingly been linked to negative effects on bees. Neonicotinoids act upon the central nervous system of insects as agonists of the nicotinic acetylcholine receptors and can have multiple negative effects on bees, such as increased mortality, altered foraging behavior, impaired learning and memory, etc. And so, as a consequence of the mounting evidence of these negative effects for bees, the use of neonicotinoids has become more restricted, and manufacturers have developed pesticides that are considered safer for bees. One of them is FluPyradiphrone, a butanylate insecticide that works in a similar way to neonicotinoids, but is from a different chemical class. Like neonicotinoids, flupyradiphrone is systemic, which means that once it is taken up by the roots or the leaves of a plant, it is translocated to all parts of that plant, including pollen and nectar. Flupyradiphrone is effective against sucking pests and can be used on many crops such as citrus, cotton, soybeans, and cucurbits. Flupyradiphrone has been assessed to have low toxicity for honeybees using current risk assessment procedures. However, these risk assessments do not consider the differential potential routes of exposure specific to wild bees. For instance, approximately 70% of all bee species nest underground and could be exposed to flupyradiphrone residues in soil when nesting on farmlands. Combined exposure to flupyradiphrone and other pesticides may also lead to detrimental synergistic effects. Recently, the Ori squash bee has been suggested as a good model species to study the effect of pesticides on ground nesting bees. It is a solitary ground nesting bee that specializes on plants of the genus Cucurbita, so squash and zucchinis. In Ontario, the squash bee spends most of its life cycle developing underground. The adults emerge in June or July and remain active only for a few weeks, coinciding with bloom in cucurbita crops. The adult female squash bee builds individual nests by excavating vertical tunnels underground. Then she provisions each nest cell with cucurbita pollen, on top of which she lays an egg. The egg hatches a few days later, feeds on pollen provision, and develops underground until the following summer. And so you can see how the species is continually exposed to soil, which could make pesticide residues in soil an important threat to their health and survival. The objective of our study was to assess the possible effects of realistic exposure to two pesticides used on squash crops, the insecticide Sevento, which active ingredient is flupyradiphrone, and the fungicide Quadristop, which active ingredients are azoxystrobin and difenoconazole, alone or in combination on the Ori squash bee. In cucurbit production, Sevento is used to protect crops against aphids and whiteflies. It can be sprayed or applied directly to soil through irrigation water and is persistent in soil, which makes its use particularly concerning for ground nesting bees. On the other end, Quadristop is used to protect cucurbit crops against common fungal diseases such as powdery mildew and anthracnose. So last summer in 2020, we performed a Semifilled experiment using 10 hoop houses that were set up at our experimental research site in Lakefield, Ontario. And we divided each of the hoop houses in two with a wall made of plastic sheet in order to obtain 20 experimental units. The hoop houses were covered with bee-proof mesh and had no floor, which allowed us to plant acorn squash directly into the ground in late May. Here, I'd like to mention that this very neat semi-filled system has been developed by Susan Chen, who has recently completed her PhD with Nigel. So we had 4 treatments: an untreated squash crop, or squash crops that were treated with either the fungicide Quadristop, the insecticide Saventoprime, or the combination of the two. We used the split plot design in which the insecticide treatment was applied to the full hoop house, while the fungicide treatment was applied to the subplots or to each off if you prefer. And we had 5 experimental units per treatment. The insecticide was applied at the maximum application rate according to label, using plastic bottles with a hole in the lid that mimicked a drip irrigation system. And this was done on July 30th, 1 week before the introduction of the bees in the hoop houses. The fungicide was also applied at the maximum rate according to label 3 times during the season at 7-day intervals, starting the day the bees were introduced. And this was done very early in the morning to make sure that the bees were not foraging during application. On August 7th, we introduced in each experimental unit 5 mated female squash bees that we collected the day before on a pumpkin farm in Guelph. We identified the bees by marking them with a dot of paint of different colors on their thorax, and then the bees spent the night in individual tubes in the fridge before being released in the hoop houses in the following morning. We then started collecting data on the nesting, foraging, and motor activity of the squash bees, as well as on their survival and potential impact on crop yield. These data were collected over a period of 3 weeks and were analyzed with generalized linear mix models. This summer, we will also assess the reproductive output of the bees by catching the newly emerged bees, so the offspring, with emergence traps, and we will collect data on their number, date of emergence, sex, and body mass. We only have 10 minutes, so I'll go directly to these endpoints for which we found an effect of the treatments on squash bees. And these are pollen collection and motor activity. So for the foraging behavior, what we did more precisely is that we measured the number of pollen grains left on the anther of individual male flowers after a single bee visit. So the night before data collection, we bagged two individual male flowers, per experimental unit, and and in the following morning, the bags were removed, and we let a single bee enter the flower. We recorded the time the bee spent collecting pollen, and once she was done, we removed the anther and placed it in a tube with ethanol. And then back to the lab, we counted the number of unharvested pollen grains on each anther, and we did this 4 times in total. We found that squash bees exposed to the fungicide collected less pollen per single visit, so there were more pollen grains left unharvested on individual anthers. And that's why you can see on this graph. So the fungicide treatment is represented on the x-axis, while the colors represent the insecticide treatments. And a potential explanation for this behavioral response is that the fungicide could act as a repellent for squash bees. This is not necessarily a bad thing in that this deterrent effect of the fungicide may actually protect the bees from by decreasing the amount of contaminated pollen with which they feed their young. But on the other hand, if the only pollen that is available has been sprayed with fungicides, a reduction in pollen collection means less food to feed the young, which could lead to a reduction in offspring production. We also found an effect of the treatments on bees' motor activity. We assessed the motor activity of the bees 2 weeks after their introduction in the hoop houses, and to do that, we caught all the bees that we were able to find in each experimental unit during 2 consecutive mornings. The bees were placed individually in tubes that were marked with lines, and their activity was immediately recorded for 2 minutes. Then each video was coded, and we counted the number of times each bee crossed the line, so the number of line crossings. So for the motor activity, we found a significant triple interaction between the insecticide treatment, the fungicide treatment, and the date of data collection. So if we look at the graph here, you have on the y-axis the mean number of line crossings each being made in 2 minutes, and on the x-axis, you have the fungicide treatment, and again, the colors represent the insecticide treatment. And you can see that the data are separated by date. So if we come back to our triple interaction, what happened is that for day 1, we found no effect of either pesticide treatment on bee motor activity. But for day 2, we did find that the bees that were exposed to both the insecticide and the fungicide in combination showed increased motor activity. So the question now is, why are we seeing this, and what does it mean biologically? So FluPyradiphrone, just like neonicotinoids, stimulates neurons. So it actually makes sense that it can increase bees motor activity. A potential consequence of this hyperactivity is that it could reduce flight performance and foraging efficiency by tiring the bees out faster compared to unexposed bees. Here, however, it's not the exposure to Sublethal by itself that led to the increased motor activity, but rather the interaction between the insecticide and the fungicides. And this could be explained by the fact that azole fungicides, such as difenoconazole, which is one of the active ingredients in Quadris, are known to inhibit the enzymes that are responsible for detoxification in bees, potentially amplifying the negative effects of flupyradiphrone. And as for the time effect, you need to know that our last fungicide application was made on August 21st. So day one is actually 24 hours after the fungicide application, and day two is 48 hours after the fungicide application. It is rather common to see a delayed in time cumulative toxic effects of insecticides for honeybees, so it may be what's happening here. So to summarize, we found two behavioral effects of pesticides used in squash crops for squash bees. First, the exposure to the fungicide quadristop led to a reduction in pollen collection by female squash bees. And in a second time, the exposure to both Sublethal and Quadris in combination, but not individually, induced an increased motor activity in bees. Our work highlights the need for more thorough pesticide risk assessment that also consider potential sublethal effects for wild bees, not just for the honeybees, especially for these new and supposedly safer compounds that are suggested as alternatives for neonicotinoids. And so with that, I'd like to thank you all for your time. And if you have any question or would like to know more, please do reach out by either sending me an email or by connecting on Twitter.